going to spend a couple days on analyzing how to analyze data on the cortical surface. So this is something I've been involved with for many years, and so we have to talk about that a little bit. And what I'm going to do is just, well, first off, like why should we do that? Why is that a good thing to do? And then talk about some of the basic algorithms and data structures, which are different than when you analyze data in 3D. So what what are we starting with? We're starting with basically a 3D block of voxels. So you have you have slices, but basically you've got this three-dimensional block of voxels. That's your basic sort of data structure. So it's natural to imagine analyzing that as a three-dimensional block, and it's got it has a brain inside of it. So here's a brain inside of it. But there's a number of different reasons to try to recover the actual cortical surface. And the first one is simply that the cortex, neocortex, and also cerebellar cortex are fundamentally 2D sheets. So if you look at an animal with a relatively small brain, or small body really, because brain is by far the main thing that controls brain size is body size. That explains over 90% of the variance in brain size. So if you have a bigger body, you get a bigger brain. As you make the brain bigger, the cortex tends to get more folded because you're kind of cramming it into a smaller space. But if you look at an animal with a very small body, the brain is flat, and that's it's very nice for analyzing things going on. In particular, we know that there's lots of maps that it occupies maybe in the human, like about 50% of the cortex contains maps of some kind. And these would be like visual maps or auditory maps of different frequencies, or somatosensory maps of different positions on the body, or musculotopic maps, so different representation of different muscles, different parts of the body. So that's a good chunk of the brain. And when you're analyzing maps, as they're sort of like going around the little folds of the cortex, it's obviously a lot easier to view these maps, on the surface. And so that, brings up one of the main reasons for doing surface-based is visualization. And what is the problem? The problem is that your visual system is not good at looking at a three-dimensional block of data and trying to sort of see it in 3D. You typically see surfaces. Now, if you have like two surfaces, you can make a transparent display so you can see two surfaces at the same time. But the brain has like 20 or 30 surfaces overlaid on each other. And if you just try to make that transparent, it just looks like sort of cabbage soup or something like that. You can't really make heads or tails of where stuff is. So that's another reason. If you look at, one of the big, another big advantage is if you have, a folded brain, it's got all these cell site in it, you can only see about 30% of the surface. So if you could recover the surface, then you can blow it up and open up all the sulci. So here's like the central sulcus and the superior temporal sulcus, and so you can see everything. You can't. You still need at least two views. So you'd have to sort of turn it around to the midline in order to sort of like see what's on the midline. But that's a lot better than sort of like cutting through 100 slices. So that's another basic thing for just looking at brain data. It's just a pain in the butt to have to sort of like page through 50 slices. But what about aligning data? So one of the big things we want to do is actually sort of find the same place. So find the same place. And one way of doing this is to line up brains on the basis of where the main folds are. Now you can do that in 3D also. So there's a method called it tends to be sort of called Talleracking after Talleyrand is this French guy, neurosurgeon, who devised this system originally for essentially trying to get a coordinate, an XYZ coordinate system for the brain. So you could go in and say, if you're trying to lesion the globus pallidus in order to release some excess inhibition from the globus pallidus, and you want to be able to sort of hit the globus pallidus, you need some kind of coordinate system. And so basically what this does, the original one was pretty complicated, but it's often done with a 4x4 matrix as an affine linear transform. And one of the problems with this, the original Talleyrand actually had like three 4x4 matrices. And what can a 4x4 matrix do? It can rotate and scale, or some combination of those, and it can translate to get things lined up with each other. But one thing it can't do is bend. And so if you've got a brain, if you have one person's brain that looks like this, and another person's brain that looks a little bit more like that, that's a bend. That's not a linear transformation. So a bend like that is not a linear transformation. Linear transformation, you can sort of stretch, or you can rotate, or you can shear, like sort of turn something into a rhombus. But you can't bend. You can't sort of make this part of the brain go up and that part of the brain go down and that part of the brain go down on the other side. So that's a nonlinear transformation. So nonlinear. We'll just call it a bend. So if you could somehow sort of squiggly morph a brain surface into alignment, you can do things more like bending. But what's one of the reasons why, even if we ignore bending, it's hard to align brains? And an example is what happens with major sulci. So if you look at one brain, here's one brain, and here's another one. And let's just take something like the superior temporal sulcus. So the superior temporal sulcus, in some brains, the superior temporal sulcus kind of stops, and then there's another sulcus behind it like that. And then you'll find another brain where the superior temporal sulcus goes like that, and then it continues up. And then, other brains, the superior temporal sulcus goes down, and then there's maybe a sulcus up like that. So what's the problem? Well, if you look at this part of the brain, what's going to happen when we compare different brains to each other? What you're going to see is we're going to have to somehow have a system that's capable of mapping something like this to something like that. So, this point needs to be mapped, to here. This point needs to be mapped to here, but this point needs to be mapped there. Now, of course you can't do that with a linear transformation, but you could do a more complicated nonlinear transformation in 3D. But the problem is it's hard to control because you've got to turn this type of a brane into that one, you've got to move this position that was in the sulcus in one brane a large distance, but the parts of the brane that are nearby are not going to move hardly at all. And so you got to kind of turn the brane into taffy in order to do something like that in 3D, and it's pretty hard. It's pretty hard to control. Of course, if you make the brane extremely flexible, you can turn any brane into any other brane, not a problem. But that's one of the problems that makes it easier to do something like this with a surface, because what you could do is you could inflate these branes, and then once you've just got the surface to line up, you can use sort of where the sulci used to be as a method of aligning it up, and I'll talk about that next time. But there's another problem, and this is the problem I call crinkles. That's the technical term for it, crinkles. If you look at the way, if you look at something like the sulcus, the superior temporal sulcus, it doesn't look straight like that. What it actually looks like in real life, it's got a bunch of little crinkles in it like this. So if this is, if here's your superior temporal sulcus like that, and we'll draw the other, so that's one bank of it, but here's another, here's the opposite bank of it. It comes down like this, and then there's the bottom of it. So, you have something that sort of looks like this. So it's got all these additional little crinkly things in it. And it's not uncommon for it to look like that. And so why is that? It's basically because how does the brain, form? So these are the crinkles there. So how does the brain form? It basically, the way it works is the white matter is pretty hard. So, when you look at the white matter, you've got this kind of hard armature of the white matter. And then what happens is, as the cortex grows, the cells in there multiply, and what happens is it starts to sort of bulge up. That's what causes sulci, basically. That's what causes the folds. So as the cortex expands against a relatively hard white matter, it's thrown up into these folds. That's actually the physical mechanism. It's likely the physical mechanism that causes this. And that's okay for forming something like the sulcus, but then it just continues to grow. And so what happens is you get all these idiosyncratic crinkles between different branes. So they're completely different between different branes. So and I saw this early on, like I was taking human brain specimens and unfolding them physically. And if you open up a sulcus like the STS, you'll see it's got all these little, hundreds of little crinkles in it, so a second level of folding. And so that, when you're doing stuff in 3D, and those are not likely to be associated with any particular functional thing. They're just different in different people. So that's another thing, trying to deal with the crinkles. So basically, how do we deal with this problem? Well, then we've got to reconstruct the surface, and to reconstruct the surface turns out to be kind of a little bit of a pain, but it's most of the algorithms for reconstructing the surface out of 3D work more or less the same way. And so there's only like so many ways to do this. And so we'll just go over some of the basic things we need to do in order to reconstruct the surface. And one of the first things, so we still, even if we're on the surface, we still might want our standard XYZ, coordinates. So that's not uncommon too. So we might, we might still end up, doing some kind of, 3D, Talarak kind of alignment, because many people still use those 3D coordinates. But if we want to actually get the surface, one of the first big problems, especially when we don't have quantitative scans, is normalization. And what's the problem here? Well, the problem is like the scan brightness might vary by like 50%, but we're looking for like a 10% difference between the gray and the white matter. So that can be a problem. So there's all different kinds of ways of trying to do normalization. We could try to collect a quantitative scan, but most people don't do that. So one example is something called histogram normalization. And that basically works by, if we just do a histogram of the brightnesses in a little chunk of the brain. So this is something that your eye and your visual system can do extremely well. So if you look at an image of a brain that's got a big brightness cast over it, like a 50%, 80% increase in brightness across the image, you'll have no problem recognizing where the cortex is. But to try to implement that into a computer program is a problem. So basically this is just brightness of the voxels, and this is just count, just number. So if you just take a little chunk of the brain and just make a histogram of it, what do you get? Well, you ought to get, if this is say 0 and 255, something like that. So you ought to get a bunch of black. So there's a whole bunch of black, and then there ought to be another peak that's gray matter and a second peak that's white matter. Now it never looks that nice. And then you've got some blood vessels up here, real bright blood vessels. So on a good day, you should be able to see, this guy should be, gray matter, and this one should be white matter. They won't be that close to each other. They won't be that far apart from each other. They'll sort of look more like something like that. Now, what happens if you go to another part of the brain that's brighter, another chunk of the brain? Then what you ought to see, there should still be black, but then you should see these peaks should shift over like that, and so you can tell from the peak approximately what is gray matter and white matter. So you basically just make a histogram of small chunks of the brain and then look for peaks in it. And then once you've found those peaks, then you can sort of shove them back into alignment, and then you'll have. So that's just one of many different methods for trying to normalize things. Those colors weren't good. Let's stick a red in there. So here's that other peak, like that. Yeah, so it shifts the peaks. So that's, and then once you've shifted the peaks, then you can do some kind of threshold to try to figure out like what is gray matter and what is white matter. So after we've done that, then we face another problem immediately, which is, how do you get rid of the skull? Because if we look at the brain, again, like, just a human looking at the brain, no problem. You can easily see where the brain is. But how do we actually get rid of the skull? And what are we talking about sort of getting rid of? So if I'd make a nice big drawing of it. Yeah, make sure it's big enough. So here's the air. Here's the air out here. So that's black. I'm drawing it in sort of light color. And then the next layer is the skin. And then we've got a layer of fat, a little layer of fat. So basically this is, and the layer of fat is pretty bright too. So this is skin, and fat. Then we get down to bone, and the bone. Maybe I should leave this. This is actually dark, so I'll just leave it dark. So the skin is light, but the fat is brighter. And then we come to the outer table of bone. So this is, and that's black. Outer table bone, and then there's the marrow of the skull. So there's another sort of light layer. That's all the blood vessels inside the skull. So you can we'll call that the marrow. And then you have the inner table of bone. So then there's another black layer, inner table bone. And then sometimes you can see the dura, which is like a little thin, leathery. So here's the dura that shows up sometimes. And then, so there's some more black. There's some more black, which is the cerebrospinal fluid. And then you have the actual cortex in there, like this. And so there's all that junk. How do we strip all that junk off? So, and then the cortex again is, the white matter is pretty light colored, and then the cortex is 10% darker. So it's 10% darker. So the CSF, that's the cerebrospinal fluid. That's black. So how do we deal with that mess? So one idea is to use a deformable template. So that's another sort of computational idea. A deformable template. And so we know that the cortex is basically, each hemisphere is basically a sphere. It's kind of like, it's not a complete sphere because it's attached at the middle, but it's basically topologically mostly a sphere. And so if we start off with a sphere and then deform it, then that's a way of basically guaranteeing that we've got the right topology to do the trick. So this is gray matter, white matter. And so how would you do that? So you would start off by making a sphere, but like a discrete sphere. So like you typically would take like a geodesic dome. Like a geodesic dome has 20 it starts off with an icosahedron, which has 20 triangular faces, and then you subdivide those triangular faces into sort of an arbitrary. You can subdivide each triangle into 4, and then each of those into 4, and keep going, and then promote the position of those subdivided triangular vertices to a sphere. So you end up getting an approximation of a sphere. So you can start off with something like that, and then try to get it to find that black layer there. Of course, the problem is another black layer there, and another black layer, and then another black out here. So all of those, the air. So the air is black, but the skull is black, and the inner table of bone is black, and the CSF is black. But if we started at the middle, so I'm just going to draw it in 2D. So let's put a surface in the middle like that. And here's the vertices in the surface, which is kind of like the corners of the surface. The actual surface is a bunch of, if you look at it, it's just a bunch of triangles like this, sort of stuck together. So you could imagine, it's like a computer graphics, typical computer graphics representation of a surface. And so what do you have? You have like, you've got a face here, and then this guy is a vertex. And so here's all the vertices, and that's what I've drawn there. All the vertices of that curved two-dimensional surface, or spherical surface. So now we've got to deform it. It's a deformable template. So how do we deform it? So what we do is we take a core sample like this. So if we go perpendicular to the surface, there's a way to calculate a perpendicular to even a curved surface. You have a curved surface, you can sort of calculate what the perpendicular at a point is. So calculate a perpendicular and then take a bunch of core samples down into the surface. And what are we looking for? We're looking for black. So none of those are black because they're all in the white matter. White matter's pretty bright. So none of those are black. And so what we do is we set up an algorithm, and it's got a couple parts to it. So one part of it tries to keep the surface from getting crinkled up or deformed. And so how would you do that? Well, imagine you've got like a vertex here, and here's the surrounding vertices here. And then there's another one sort of coming out this way. So those are arrows going to the surrounding vertices like that. And so one thing you can do is if you calculate the normal, so here's the normal, that's perpendicular, so that's approximately perpendicular to all these guys. If you calculate the normal, then we can do a couple things. So I won't write out the equations there in the notes, but one thing you can do is basically, if you look down on the surface, try to prevent something like this from happening. So where that guy is off center. And so what you want to do is like move that guy so it's evenly distributed. So that will sort of stop it from scrunching. But the other thing you can do is stop the surface from doing this. And so in that case, what you want to do is reduce the curvature of the surface. So move this guy to sort of to make it less curved. So this is squishing. This is squishing. Reduce squishing. Sort of evenly distribute the vertices and then reduce curvature, so it doesn't crinkle up. As sort of like a blanket. So reduce curvature. So you can do those two things, but at the same time, we're looking for black. And so how do we look for black? So here's the equation for how to push on that vertex. So you can think of it as like the force on that vertex due to the image data. And so we need some adjustable parameter to say how strong the force is relative to these other forces. So we've got number 1 here, we've got number 2 here, and number 3 here. So this is a knob to say how strong should we make this force relative to these other ones. But then so this is our our image force. So then we'll multiply that by the normal of that center center vertex. So here's that normal. Here's the n right there. And what are we going to multiply that by? So we'll multiply that by this symbol, which just means sum up everything to the right. Multiply everything to the right, and then get the sum over like a whole bunch of distances. Okay, so, and what are those distances would be, going down, ticking down here. This is like d equals 1, d equals 2, sort of going, ticking down this little core sample. And so what are we summing up? So we're summing up the maximum of either 0 or the tanh. So that's like a sigmoid. So what is that hyperbolic tangent look like? Let's just draw it out. So that guy, it looks something like this. So it goes up to, it goes from -1 to 1. It goes through 0 right there. So if we feed in something as an argument to that, we'll get something that sort of goes between -1 and 1, so it doesn't go completely off the wall. And then what are we taking the the inverse, not hyperbolic, inverse tangent? So this is this is tangent. And this is x. Okay, so what we're going to do is take the image value. So now we have to sort of put an image in here. So, what does an image look like? Well, there's going to be a bunch of voxels here. So what color should we draw them in? How about green? So here's the actual voxels now. Okay, and each one of those has an image data in it. So some brightness, equals, brightness. And so what do? We go to a point. So it's go to a point which is the coordinates of our center vertex. So this guy right here. So that guy has some coordinates. And what does that mean? Well, that just means like, x, y, z of the vector. And what is the vector? Well, the vector is, wherever the center of the coordinate system is. Okay? So here's 0 in the coordinate system, say in the middle of the brain. And this guy is, the vector that goes from the center of the coordinate system to the center vertex as opposed to its neighbors. Okay, so that's what that R means. Get rid of that. Okay, and what does I of R mean? I of R just means whatever the voxel that 3D coordinate was in. So you can see here's that 3D coordinate. It was in that green voxel that I outlined, and so that's the image, that's the I, the brightness of that particular voxel. But now we're going to subtract off from that the whatever distance down into the core sample we are times the, that surface normal. So what we want to do is first, find out what voxel we're in here, then go here. In that case, we're in the next voxel, and then we go, two times the surface normal, and then we're in this voxel. So what this does is it sort of samples down the core sample, multiplies them all times each other, and we're almost done. And then we subtract off the image value of the target. And what's the target? The target is black, or something that's kind of dark. Okay, so here's the parentheses for what we're taking the tangent of. And then here's it is hyperbolic tangent. Yeah, hyperbolic tangent. And that's what we're taking the max of. And so now what does this mean? So this means, this is, target, for example, dark, like say, like 30 might be a dark level. And so if we're not dark, when we subtract off dark, we're going to get a positive number, and that's going to make sort of a positive number over here. And then if we take the maximum of that, we're going to get that positive number. But as this surface, and so what will happen is there'll actually be some push on the surface to sort of move it, meaning like we're not dark, so let's expand outward. So this thing is, you can think of this as the force on it. So it'll expand outward as long as it's not dark. So this is, outward force. And then as soon as it gets dark, as soon as one of these vertices gets dark, then doesn't matter which one, as one of them gets dark, since they're multiplying all times each other, and we're taking the max of either 0 or a tangent. In other words, like as soon as it's going to be 0 as soon as it gets. So this this max makes that happen. So it'll be 0 as soon as the value of the image voxel gets less than the target voxel brightness at 30. Then that will turn off the force. And so what will happen is, and this is just for one vertex, but you do it for all of them at the same time. 1,000 of them at the same time, a couple thousand of them at the same time. And so what will happen is the surface will this deformable template will just deform. It will sort of like move outward until it reaches black. And then as soon as it reaches black, it'll shut off the force. And so then you'll end up with a little surface. It won't go all the way down into sulcus because it's trying to, not be squishing and not have too much curvature. It'll go down a little bit. And so you'll end up with something that's right on top of the brain. So that's an example of, some of the things. So this is end. This is start. So now, on a computer, you can do that. It takes like a couple seconds. And then you can quickly strip off all the rest of the stuff that's outside here. So that just sort of gives you some idea of the kind of... Now, you might think, well, why don't you just like do it manually? Well, it takes longer than you think. So you'll end up with like 200 slices, and you've got to edit 200 slices. So you could do that, but if you sort of want to do a reasonable number of brains, that will end up taking weeks. And so, whereas you just have a couple seconds of computation. So now we've gotten rid of the skull. And now we've got to do a very important filter operation. This is anisotropic filter. Now, why would you want to, you've got like good quality data, why do you want to filter it? And the problem is, how many voxels are there in the data set? Well, like 16 million. So So the problem is, if you've got some kind of automated operation, and say it makes only 0.1% error, it only screws up like 0.1% of the time. Well, 0.1% of 16 million is a lot. That's way too many. That's way too many things to fix by hand. And so one of the things you find out is that if you try to write algorithms like this, is that you really have to be extremely accurate in your segmentation of the images. And so what kind of things can you do to help with that? And one thing is to do an anisotropic filter. And so what are we trying to fix? So what we're trying to fix is, you've got a brain image like this, and so now we stripped off the skull. So now we just have the gray matter. So that's all gone. But when you look at the gray matter, if you look at it as a human, here's all the voxels. There might be like one light voxel in a sulcus. And so here's the gray matter, which is light, and then the sulcus is dark. But there's just like a stray little light voxel there. And if you just looked at the image, you wouldn't even notice it. You'd say, yeah, it's whatever. It's, I can see where the sulcus is. But if you're trying to classify that as white matter, what will happen is, and say you're trying to make a surface. So your surface will, come along like this, and then it will hit that light voxel and go like that. And then you'll have another little piece of the sulcus attached underneath it. And so you'll have the heartbreak of a wormhole, little wormhole between the sulcus. And so it turns out, if you only had like 10 of those, you could fix it by hand. But what if you had like 10,000 of those? It's just impossible. It takes too much time to go through the data set and that 16 million voxel data set and fix it. So you actually have to sort of like be really good and get rid of those things. And so one way of doing it is, you could smooth the data set. And so how would you smooth it? You would do something like a 3D Gaussian. So convolve with a 3D Gaussian, sort of blur the 3D data with a 3D Gaussian. And that would fix this problem, because all the surrounding voxels in the sulcus are dark, and so it would darken it down a little bit. But the problem with that is that would blur the data set. And in particular, say you have like, here's gray matter and then here's white matter. What happens when you do a convolve with a Gaussian? You'll get something that looks, like something like this. It'll blur it out a little bit. And if you're now thresholding, say like, I'm going to define that as the thing that means that I'm in white matter, it will have the effect of actually moving the boundary of the white matter. You could try to sort of select this as the white matter, but then maybe you would get some other junk and accidentally misclassify something as white matter that was actually gray matter. So blurring the data blurs it a lot. And so what we could do instead is, we could 2, 3, 4, yeah. So what we could do is take a little chunk of data. We don't know where the surface is yet, so we just take a little chunk of data like this. A little sort of 5 by 5 chunk of data, and go through it and try to find out what is the plane of least variance. So we know that we're dealing with something that has like a surface in it, and so that you can think of that as a prior. So imagine just going through this little chunk. You don't know where the surface is, but just go through it and find out which plane in there. So here's a plane in there. There's a little plane in there. Has the least variance in it. And then only smooth in the plane. So, only smooth in plane. And so that that will blur the data a lot less, and it will fix something like that. It'll fix those little speckly problems. And if you don't do this, it's like I said, it's just like there's way too many voxels to fix by hand. You're not going to fix 10,000 examples of that. So some kind of filtering is pretty critical. But we still don't have a surface, and so then how do we make the surface? We make the surface by, flood fills, something like the 3D. It's like the paint bucket in a paint program. And so, how does the paint? If you pour the paint bucket, right? So here's the paint bucket, sort of with stuff pouring out of it. How does that work? It just tries to find pixels in 2D. So if you if you've got something like this, and there's something like that, you put the paint bucket in there, it will basically just fill up anything that has pixels that look similar. And that's the principle, except we just do it in 3D. You can do the same thing in 3D. But there's some problems as usual, so you have to do it a couple times. So 3 times, actually, as the case may be. So what happens is we're trying to find the surface, and so the way we find the surface, so here's the brain in there, and there's some junk outside, and then there's like a ventricle inside. So that's let me draw that for all of these. So there's some bright things outside. Maybe your skull strip didn't work exactly correctly, and you've got a little bit of the skull sticking out. That happens all the time. Okay, so what we can do is start off by putting a seed in the middle, and then in 3D fill up anything that looks similar. So you'll basically sort of fill up a block of mostly white matter, but there's going to be something, you're going to have a ventricle in there. And so if you're saying that I'm going to define the surface as the boundary between filled and not filled, well, then there's a piece of the surface in here too that we don't want. So that was our first seed. So that now this guy sort of disappears because it didn't get filled. So this guy is gone. But how could we deal with that one in the middle? So what we could do is put a seed out here. So now I'll just fill here, and what are we going to do? Well, we're going to, unfortunately, fill around that guy now. But we have the effect of getting rid of this guy. So that's a way of getting rid of that one. And then how do we get rid of that outer one? Now that we've gotten rid of that one, put the seed back in the middle like that, and fill up. And then we'll finally just get a complete block of voxels in which the boundary between filled and unfilled is the surface. But it's not the surface yet. And so what we have to do now is to actually make a surface out of it. So for that we have to sort of tessellate the surface, figure out where the boundary between filled and unfilled is. And how do we do that? We have this giant pile of voxels, and we go to where we see a boundary between filled and unfilled, and what does that look like? So it looks like a bunch of cubes, right? So here's those voxels. So there'll be a whole bunch of cubes, just a giant pile of cubes of 16 million cubes. So here's another. Make sure I draw enough of these. So there's just a giant pile of cubes like that, and wherever we have a filled cube, we know that that's a boundary between, that's where the surface is when there's a filled cube next to a not filled cube. And so now what we have to do is figure out, the neighbors, and you notice there's different numbers of neighbors. So that, that guy has 3 neighbors, but this guy has 4 neighbors. So basically we put those, put that surface, we essentially just make a giant table of who everybody's neighbors are. And this is the way all surfaces are represented in any kind of video game. If you're looking at like somebody's face, you have a mesh, which is a triangular mesh, and then the mesh knows who all the neighbors are. Now, the thing about that is, initially we were starting off with a 3D coordinate system, and the 3D coordinate system, was basically just x, y, z. So, each voxel just has an x, y, z position, and it's relatively straightforward. What we're transitioning to is a 2D coordinate system. So this was just, a 3D matrix, a simple 3D matrix, but this is no longer a matrix. It's actually a list. And the way that this data structure looks like, it looks like what's the minimal amount of information you need to store in order to make a surface. And the first thing you need is a vertex list. So what is a vertex list? Why bother numbering it? So here's the first one, just leave that out. Here's the second one, just leave that out. We just say we know that save space by not writing down the vertex number. And then here's the XYZ. So, that's XYZ 0. Here's, X1, Y1, Z1. So you just have just a list of coordinates like that, and those are the vertices. So that's the points in the. But we need another thing if we're going to draw it, and actually, you could just draw that as a bunch of little dots, but that doesn't give you a very good idea of the surface. And so then we need a separate list of faces. So we have a face list, and this is the way like I said, any kind of 3D graphics involving surfaces works. So we have a face list, and so the face list, we could also, don't bother numbering it, because we'll just assume that they're in particular order. And then we have the numbers of the vertices. So the numbers of the vertices. So here's one vertex, and that might be vertex, 12,421, something like that. So that's that vertex number. And so what's in the face list? Vertex numbers that we didn't include over here, right? So it's the vertex numbers of the neighbors. So there'll be like, A0, B0, C0, D0. So these guys are vertex numbers. And then here's the vertex numbers for the second face. 1, A2, B2, C2, D2. Okay. So that's actually how that's how a surface like in FreeSurfer or any kind of animation program is represented. Now, there's not enough information there, so when you read that surface in, the first thing you have to do is you have to find the neighbors of each vertex, because it's not specified there. It's only what's specified there is the vertex numbers of each face. But who's the neighbor of this guy? Well, we've got to figure that out. So find neighbors. And the other thing you can do is calculate normals, surface normals. And those are very critical for a whole bunch of different things. If you want to show the surface, display the surface at all, you need the surface normals. And why is that? Well, if you just draw a brain like that with no lighting, it just looks like a flat piece of cardboard. It doesn't look like anything. And so in order to actually make it look like a brain, we have to sort of define a sun somewhere. And then we just look at what the angle to the sun is compared to the surface normal. So here's the surface normal, surface normal to that part of the brain. And so that one is sort of more or less in the direction of the sun, and so it should be pretty bright. But if we've got something down here, that's normal to the surface like that, then that guy, and we look at the vector going to the sun and take the dot product. Remember the dot product. Dot product will say, like, are the vectors similar? So these vectors are kind of similar. These ones are kind of opposite. And so you can control the brightness. And then what will that do? Then you can shade this part of the brain brighter and this part of the brain brighter, or the top of the, and then you'll actually get something that, that actually looks like a brain. If you don't do that, it just looks like, it's amazing. It just looks like a piece of cardboard, and you got no information. So that's one thing you need. If you want to display anything, you need that. But then you also might want to use the normal for doing computations, like deforming the surface. And so there's a lot of things you could use the normal for. But what is the most important point about this? So we started off with something that was just XYZ, and so you just have like a functional activation at a particular XYZ coordinate. Very easy to deal with. In this case, now what are we doing? Well, we've got two different things. So if you want to display the data, we use the faces, because you don't want to just draw the vertices, you want to actually draw the faces. And but what is the data? The data is associated with the vertices. And so what are the things that are associated with a single vertex here? Well, there's a whole crap load of things. So the very first thing, of course, you've got the XYZ. But say we're moving the surface and we want to sort of see how fast we're moving the surface. You could have, last XYZ. In practice, you end up with hundreds of things associated with each vertex. So, that's the last XYZ. And then we calculated the normals. So the normal X, normal Y, normal Z. And then we've got some statistics. Maybe we calculated an F ratio. So we've got an F ratio for this voxel. And then we've got we might have a time course, like a whole time course with that voxel. And, then, so there'll be literally hundreds of things associated with that vertex. Or we might have local surface coordinate systems. There's a whole bunch of different things. So how do we display it? Well, so now you have to think about how you display it. Well, you've got, here's, a little chunk of the surface. So they're little. So usually what we do is we start off with the surface defined as rectangles, because that's the face, the face of the original blocks, little cube blocks. But usually what we do is we divide these into triangles, because they're easier to deal with in triangles. So now we've got them divided into triangles. But how do we display that? We display that by, say you've got a statistic like an F ratio. Well, there's going to be a different F ratio at this vertex and this vertex and this vertex, and so we have to interpolate. When we're filling in all the pixels that fill up that. So you're always going back and forth between displaying the data, which is actually present at each vertex, but then you have to display it as a face. So you can sort of see why a lot of mathematicians would say, that's an ugly thing. I would rather deal with this. It's just 3D. Everything is just on a straightforward grid. It's much easier to deal with. And last but not least, how do you actually, get data onto the surface even? That's a whole separate operation there. So if we look at our reconstructed surface, so let's say we've reconstructed the gray-white matter surface. So here's white matter and gray matter. So here's our surface, the vertices in our surface. And so they'll be pretty close to each other, usually less than a millimeter apart from each other, because we made this surface out of an MP rage that was a high-resolution surface. So there's all our vertices, but we did some functional MRI, and so here's some functional MRI voxels. They're bigger. They're maybe something like about this big. And we lined them up. We lined up the functional MRI with the structural scan, so that we have, that's another whole nightmare, but how do you do that? You have to align up that functional data with the actual structural data. But say we've got it lined up, and then what we do is we can say, okay, what's the data at that vertex? So one thing we could say is, maybe the data at that vertex is whatever voxel it's inside. So this is fMRI data and stats. So we might have had a time course, and then we calculated an F ratio out of the time course, and so and we have one of those time courses and stats at each voxel here. But what are we going to say? What is that voxel? What does this vertex actually get? So we could just say, well, maybe it gets the one that it's inside, but in that case, it's going to be kind of close to the gray-white matter border. And so instead, what we could do is get the surface normal that we calculated, and then go up along the surface normal, and say we'll go like halfway up the surface normal until we hit the peel surface, and we could say whatever voxel you're in halfway up is the one that we want, and so that might be this voxel instead. And then we could take that stat value. This is 3D, 3D fMRI, and we could take that stat value and stick it into this vertex, into this data structure. And so now we'll have that vertex, the F ratio will be in there. So you can see it's a kind of a bit of a pain in the butt compared to 3D. In 3D, you just got you just did your analysis. You say this is gray matter, XYZ, and this is stats, XYZ, you're all done. Here we've got this more complicated thing where we've got to sort of sample the data onto the surface, and you're dealing with a list. So this thing here is your surface specification. You're dealing with a list rather than like a simple 3D matrix. But there's a lot of advantages to it because the cortex actually is a surface. It isn't just a 3D block of voxels, and so there's a lot of extra information you're adding here. And as we'll see, you can use a surface to do a better alignment across brains than you can do by doing things just sticking in 3D. And you can much more easily visualize, where the activity is in the brain instead of trying to cut through in sections through the folded surface. Okay, so that was introduction to the nightmare of using surfaces. But like I said, there's a lot of software out there which me and other people have written to sort of automate this process. But it's good to know what's actually underneath the hood when you use the software, because a lot of things aren't defined in it. Okay, any questions about surface-based? Well, next time I'll go into more detail about how some of this stuff works and how you can align brains using the surface. Okay, surface lists. So if you download FreeSurfer, this is what it actually actually does. Okay, I guess we'll end the overly long recording there.